Hi, everyone. This is an introduction into the scientific method exercise for the first week, August 24th. Let's begin. We're going to go over the scientific method not because we don't think that you've come across it before. It could be that you have come across it, but it was a long time ago. But the real reason why we go over the scientific method is because there's a couple of areas within the scientific method that don't get discussed in richer detail. And we want to be able to do that for you all in this exercise. So let's first begin with listing the steps of the scientific method. We all know that everything begins with an observation. We take notice of some natural phenomenon, it piques our interest, and then that usually leads to some sort of question. Or it could lead to a series of questions. And I think that it's our natural intuition to try to answer our own question. And we do that with something called a hypothesis. A hypothesis is supposed to be a tentative explanation. It's not meant as like, okay, this is the answer. It's a tentative explanation, and it's one that we need to challenge. We need to test and see if the hypothesis is supported or if it is rejected. 4 is a prediction. Once we have our hypothesis, we then make predictions. Predictions are usually in an if-then format. If the hypothesis is true, then this should be the outcome of whatever. That's the prediction. And number 5 is the test/experiment. Oh, what's going on with my penmanship here? Every test or experiment should have a control. Without a control, you cannot draw conclusions about your experiment. We're going to see that in the later part of this presentation. Okay, it's the question and the hypothesis that I want to make a big deal out of in this presentation. This is the part that isn't really talked about in rich detail, I feel, when the scientific method is discussed. So let's look at the question in a little bit more detail. For the scientific method to work, a question must be well defined. And what that means is that you cannot have ambiguous concepts, terminology. We all need to know what exactly you mean in your question if we are to come up with a hypothesis and eventually test for getting clarity on your question. Questions should also be testable. It does us no good to ask a question if we cannot ever get to the part where we actually test it. And if we can't test it, then there has to be something that we can measure, something that we can record. Your question should also be controllable. Here's what that means. In every experiment, there should be some sort of control, and there should be an experimental group. The control group and the experimental group should be identical in every way, except the one thing that is different between the two, the thing that you are testing. That's what we mean by controllable. I'm going to give you an example in the later part of this presentation. And I hope that by then you'll understand the difference between a controllable variable versus a control group. Those are two different things. Finally, a question should be reasonable and consistent with existing bodies of knowledge. Again, it does us no good to ask a question if we cannot get to the part where we test it. If we don't have the technology, if we don't have the background information, we may not be able to carry out an experiment. So at that point, it does us really no good to ask those questions. Okay, let's look at some examples of questions, and then you decide if, or we decide, if they're good or not. If they're good enough to get us through the scientific method. Are serial killers evil by nature? Most students would have an issue with the word evil. It turns out that evil, some groups might consider some actions to be evil, whereas other groups might consider them perfectly acceptable. So we don't have a consistent consensus agreement on what is evil. So evil needs to be defined. I might think I know what evil is, but I might also discover that I don't share the same ideas of what evil is with others. So just for my own understanding, I would need the person who originated this question to tell me what they mean by evil. Evil as defined by what? Another issue some students point out is the word serial killers. They may not know what the definition of a serial killer is, and they would like to see it somewhere in the question. We can go to an FBI website and adopt their definition if that's what's required. Let's look at another one. What is the cause of AIDS? That's well defined. That's testable, measurable, controllable. And the answer is it's mostly attributed to HIV, a virus. But there are other ways in which a person can develop AIDS. It's not just by way of HIV. Why is grass green? That's a good question. It's well defined, testable, measurable, controllable. And we should all know why grass is green. And if we don't, we're going to learn about it this semester. What is the best recipe for chocolate chip cookies? Most students would jump on the word best as being subjective. It's true. I might like some chocolate chip cookies that you don't like. And you might like some chocolate chip cookies that I don't like. And it could be something about texture. Soft and chewy versus crumbly and crunchy. I don't know. I know that the people from the East Coast don't necessarily care for our In-N-Out burgers. I think it's because they're weird. But then I'd have to define what weird is, right? I think you get what I mean. The word best is subjective. But I want you to understand that there's all kinds of cook-off competitions. I particularly like the grilling competitions for who made the best brisket and tri-tip and ribs. And there's a panel of judges, and they've come up with a criteria for what qualifies as best. It includes things like color and texture, smokiness, the flavor profile, the right amount of heat and sweet, and so on. Salty goodness, fat. You get what I'm saying? There's an effort, in other words, to describe what best is, right? To come to an agreement on what is best. And then they can be as fair and impartial as possible in determining who had the best brisket, let's say. Also, did you know that the good people at Ben & Jerry's hire food scientists? Guess what they're doing? They're trying to come up with the next best ice cream flavor. They look at things like data that shows what is it that people tend to like the most. What combination gives you the right activity and taste receptors and so on, and makes you feel all warm and fuzzy inside. See, there's an effort there, right? A scientific effort to determine what's best. All right, let's look at the next one. When will the big earthquake hit San Francisco? All right. Listen, I don't know. I mean, if you're somebody that lives in San Francisco, there can't be too many more important questions to ask than when will a big earthquake hit, given the fact that they're in California and that San Francisco has a rich history of big earthquakes. Not long ago either. And yet, we don't have the technology to be able to tell them with any kind of certainty about when a big earthquake will hit, let's say a month from now, which gives us some sort of time to mobilize people and try to mitigate the sort of infrastructure damage, the injuries and death toll. We just don't have the capability of performing a test to figure this out yet. So this would be an example of failing maybe as a reasonable question consistent with what we know. We just don't know enough to be able to answer this. How can the maximum yield be obtained from a peanut field or any crop field? It's a good question. It's well defined, it's testable, there's measurable, right? The measurable part would be how much of something you produced. All right, so I hope you're understanding what it is that we would look for in coming up with a better question for the scientific method. Let's now look at a hypothesis. A hypothesis should be testable, just like the question, and perhaps even more importantly, it should be falsifiable. You cannot design an experiment to demonstrate that your hypothesis is correct. You need to challenge your hypothesis, You need to be rigorous against your tentative explanation. This is not the time to design an experiment to show that you are right, to confirm your biases, or to confirm your gut feelings about things. This happens all too often today in society, right? You need to challenge your views. You need to challenge your ideas. You need to challenge your hypotheses, your tentative explanation for things that you observe, questions you have. You need to challenge those. That's doing science. If you design an experiment with the intent to show that you are right, you're not doing science. You're doing pseudoscience. That's fake. It's fake science. All right. And it should be written as a statement. Okay, let's look at some examples. Plants absorb water through their leaves as well as their roots. I imagine that the question was, I wonder if plants absorb water through their leaves, or is it only their roots? The hypothesis here is, I think they do. I think that they absorb water through their leaves and their roots. The answer is actually interesting. The answer is that the majority of plants absorb water through their roots, not their leaves. In fact, you don't want to water leaves in many cases. But there are a small group of plants that descended from the most ancient primitive land plants. We call them bryophytes. You know them as mosses, and they grow in damp environments. They have to. They don't have roots. So they need to be in moist environments where they can absorb water through, you guessed it, their leaves. So that's an interesting answer. Next one, mice require calcium for developing strong bones. That's a good hypothesis. It's written as a statement, it's testable, and we can falsify it. I think you can imagine how to do this test. Take a bunch of newborn mice, split them into two subgroups. One group gets calcium, the other group gets no calcium. And then when they become adults, I don't know, we can scan them, we can x-ray them to determine their bone densities. And I think what we would find is that you kind of need calcium for developing strong bones. So that's a good one. Dogs are happy when you feed them steak. All right, I have dogs. I've had dogs for a long time. I think I know when they're extra happy over something that I give them to eat. We call them treats. They're treats for a reason. They get extra hyped up and we like that. But I don't know that I can tell on all dogs. So we need a measuring tool or system that's going to be reliable, objective. So I think if you had asked me this question 20 years ago, I'd be uncomfortable with, I don't know that we can test for that and get an answer that we can, be comfortable with. But today, I know that dogs are subjected to MRI machines. And then they're exposed to a variety of things, visual cues, auditory cues, scents. And then we look at their brain scans and we see what parts of their brains light up. We can tell if their happy centers get lit and to what degree. So today, I think we can answer that question a little bit better than in the past. An active volcano can be prevented from erupting by tossing a virgin into it during each full moon. I know what you're thinking. This is horrible, but you're laughing. Okay, I get it, it's silly. And there is no ethics panel on any academic institution that's going to approve this experiment, right? Academic institutions have an ethics panel that take a look at your proposed animal study. This qualifies as an animal study. And yeah, nobody's gonna say, yeah, let's go ahead with this study. But as far as a hypothesis goes, it's a statement, it's testable, and it's falsifiable. It's also horribly funny. The higher the intelligence of an animal, the more easily it can be domesticated. The word intelligence is underlined. Usually that means that it's a live link. If you click it, it'll take you to a website where you can read more about intelligence. There's different types of intelligence. There's modes of intelligence. I don't know which intelligence is being measured here. Also, it's important to know what type of intelligence we're measuring because we're proposing to come up with a one-size-fits-all test. I heard ravens are smart, elephants are smart, dolphins are smart, dogs are smart. Is there a one test that's going to be fair to all those species? I don't know. Maybe you have better ideas. I'm just sounding off all my concerns. HIV can be transmitted by cat fleas. It's written as a test, as a statement. It's testable. It's falsifiable. Okay, I hope you understand that questions and hypotheses need to be scrutinized a little bit more than maybe you were aware of. Let's now talk about variables. There are 3 variables I need you to know: the dependent variable, the independent variable, and the control variable. The dependent variable is the easiest variable to identify in an experiment. It's easy because it's the one that's being measured, counted, or recorded. As soon as you know what's being recorded, what data was being written down, as soon as you know what that is, you know the dependent variable. The independent variable is whatever is different between the control group and the experimental group. This is a variable that's deliberately varied. The more difficult variable is the control variable. Again, when you have a control group and an experimental group, both of those groups need to be identical in every way, in every factor, every variable that can affect the outcome of the experiment. It needs to be identical in both groups so that there's only one thing that's different between them. And the one thing that's different between them is the independent variable. That's the thing that was deliberately varied. So let's go through these variables by way of an experiment. Here's an observation I've made over the years. The observation is that vitamin C is heavily promoted during cold and flu season. I think you know what I'm talking about. You've heard of Emergen-C, Airborne, and other over-the-counter symptom relief medications. They go out of their way to let you know that there's vitamin C in there. Here's my question. Does vitamin C shorten the duration of a cold? So I want you now to apply the criteria to this question. Is it well defined? Is there anything in this question that is not clear? Is it measurable? Can you imagine how I can do a test? I think so. It's a good question. Now our hypothesis. The hypothesis should not be written like some sort of elegant thing. It should be straight to the point. Here I'm going to say vitamin C shortens the duration of a cold. I could have also said vitamin C does not shorten the duration of a cold. Regardless of how I wrote my hypothesis, I need to ensure that my experiment challenges the hypothesis. Okay, my prediction. Those taking vitamin C, or those with vitamin C, will have the cold a shorter time. So again, I just kind of made this short, but it should be written as an if-then statement. If you take vitamin C, you should have the cold for a shorter period of time. Okay, and what's my test / experiment? Again, always with a control. It's easy, right? I think you can imagine it. I'm going to have 2 groups. One is a control group. One is the experimental group. One group is not going to receive no vitamin C, and the other group with vitamin C, they're going to get vitamin C. That's the only difference between the two groups. That's the only difference. That's the thing that I am varying. And for that reason, vitamin C is our what? It's our independent variable. What am I measuring? Because that's the easiest variable to identify, the dependent variable, right? So what am I measuring, counting, or recording? It's the duration of a cold, and that's in days, right? So this is my dependent variable. And vitamin C is the independent variable. But now I need to come up with my controlled variables. And in your exercise for this week, you need to come up with 4 for whatever experiment you do, whether you do the figuring out the source of a sound, figuring out how long you can hold your breath before or after exercise, or the temperatures of different parts of your skin. You need to come up with 4 variables. So if my experiment here on vitamin C and the cold is supposed to be a representation of the US population, then I should probably make sure that I have equal representation of ethnicities, right? I should probably have equal representation of sex. I should probably have equal representation of age. But that brings up an interesting question. Because we know that senior citizens, babies and toddlers have either a declining immune system in the case of senior citizens, or a brand new developing immune system in the case of babies and toddlers. I need to decide, do I include them in my experiment or not? If I decide to include them in the experiment, they need to be in both the control group and the experimental group. If I decide not to include them in the experiment, then they're not going to appear in any one of those 2 groups. They're not in the control group, they're not in the experimental group. But what if I did something mischievous? What if I did something where I included senior citizens, babies and toddlers, only in the control group, and I left them out of the experimental group? Maybe I have stocks in vitamin C. Maybe I want people to buy vitamin C because then I make more money. Do you see that the way I design my experiment is going to make it look like vitamin C is good for you? By including individuals with a less than ideal immune system in the control group, that's going to make the control group average amount of time with the cold longer. And because the experimental group doesn't have senior citizens and doesn't have babies and toddlers, they're going to have more of the robust immune system having individuals. And therefore, they're probably going to have the cold a shorter period of time. And it's going to make it look like vitamin C is great for you. But is it? I mean, I really want to know. I don't want to know that the experiment has been corrupted or compromised by having too many different variables. There should only be one variable that's different, vitamin C. Not age, certainly not compromised immune systems. Okay, I think you understand now what I meant by a good hypothesis should be falsifiable. And you need to show that you're trying to falsify your hypothesis by coming up with a rigorous experiment where you challenge your hypothesis, as opposed to designing the experiment to make it look like your hypothesis is correct. Okay, what other things can we look at? We can also ask individuals that might participate in this experiment, we might also need to ask them, do you have any pre-existing conditions? Do you have emphysema? Do you have asthma? Do you have any issue with your immune system already? And then we need to decide, do we include them or not in the experiment? But whatever we do, we need to make sure that they are equally represented in both groups, because there can only be one difference between both groups. And in our specific case, that's vitamin C. By the way, I know you're curious. There is no conclusive study to demonstrate whether vitamin C is good or not in shortening the cold. None, 0. But you wouldn't think that based on the heavy promotion of vitamin C right around cold and flu season. Very interesting. And I have some other nasty things to say about airborne. I might save that for lecture. Okay, let's practice identifying variables, dependent and independent variables at least. Let's just do a few. Mice are kept at different temperatures for 6 weeks. percent weight gain is recorded. Okay, what is the dependent variable? Remember, that's the easiest one because that's the one you measure. So what are we measuring here? Percent weight gain is recorded. That one's, I mean, straight out giving you the answer. The independent variable is the variable that's varied, the one that's being different, right? Mice are kept at different temperatures, all right? See, there it is. If you look for keywords in the description of the experiment, you can identify the dependent and independent variables. Let's look at the next one. A hypothesis proposes that dog weight increases with fewer litter mates. So what are we recording here? Dog weight, that's the dependent variable. And what is it that we think affects that? What is it that's varied? The amount of litter mates, that's the independent variable. Let's do one more. Seeds are soaked in salt solutions at different concentrations and germination is counted each day. Okay, what's germination? This is when seeds begin to sprout their roots and their leaves. That's germination. Seeds are soaked in salt solutions of different concentrations and germination is counted. So what are we recording? Germination, that's the dependent variable. What is it that is being varied? What is it that affects the germination? Different concentrations of salt, that's the independent variable. And so on. Okay, final comments. Whenever we design an experiment, sometimes we need to consider levels of treatment. Let's go back to our experiment. In the experiment with vitamin C, if I weigh 200 pounds and I'm in the experimental group that receives vitamin C, and I get 1 pill a day, what about a person that weighs 100 pounds? Do they also get 1 pill? Or should they get half a pill? See, that's a level of treatment, right? Level of treatment can also be something like different temperatures or different concentrations of salt. Those are examples of levels of treatment. Sometimes they're applicable, but they're not always applicable in all experiments. Replication. In my experiment, if I had 500 individuals, then I had 500 controls. If I had 500 in the experimental group, then I had 500 experimental groups. That is replication. But replication can also be another 2 or 3 scientists replicating my experiment to see if they get the same results that I did. That would also be replication. And the reason why we do replication is so that in the end, when we report something to society at large, we have what? We have confidence. The data is robust and it's compelling. And that's when it should come out to the public. And finally, every experiment should have a control because you cannot draw conclusions unless you have a control. Imagine in our vitamin C experiment that I did not have a control and all I had was an experimental group and I gave them vitamin C. And they had, on average, 500 individuals, on average had the cold for 10 days. Is that good? Is that neutral? I don't know, because I didn't have a control. So I cannot even draw a conclusion. But what if I had included a control and I determined that the control group had on average the cold for 20 days and the group that had vitamin C had the cold for 10 days? Well, in that case, yeah, the data is compelling. We should all be pounding the vitamin C because it's going to help us with the cold. Except again, remember, there's no study that shows that it helps. All right, so I hope you understand that you need a control if you're going to draw a conclusion. No experiment can be done without a control. All right. Okay, that's it. In your worksheet, you're going to have an opportunity to identify variables, to design an experiment, and if you need any help, please do not hesitate to contact me for help. All right, have fun.